So that's a negative stain. Now next, we want to spending some time talk about the differentiated stain. Gram stain, acid-fast stain. Indole stain, flagella stain, capital stain, but we were spending a lot of time talk about this gram stain first. Okay, this is usually is a major part of microbiology staining technology. Talk about the gram stain. So we want to spend in the next roughly 20 minutes, 15-20 minutes to talk about the gram stain. So let's prepare some of the color, which is blue color, red color I need, and I also need a black one. Let's do the black one. Okay, so gram stain. I believe you heard about this before, sooner later or more or less. So gram stain. Gram stain is created in 1980s by Dennis Gram, which is a Danish microbiologist, to do is a staining technology. It is very magic. It can be differentiated basically bacteria into two categories, which is gram positive and gram negative. Based on the staining results, gram positive stains blue or purple. Some people say blue, some people say purple. I usually say purple. Gram negative bacteria, it stains red or stains pink. But how about the fungi? Yes, we can do it. For fungi, we basically say gram stain blue, just simple. Not positive or negative, we just say gram stain blue, okay? So what are the gram stains and major steps? Why it is so magic? Gram stain have 4 major steps. Now obviously you have to prepare every these smear preparation. Put a drop of the water, draw a circle, put the bacteria there, air dry heat fixing. But after heat fixing, we need to do the procedure to add the dyes there. So what are the major steps? Number 1, crystal violet. Crystal violet is a very purple dye. It's a strong positively charged. So the function is you're going to add dye color on the surface. The second step, we're going to add gram iodine. Little bit of brownish color. What's a gram iodine's function? We call it a mordant. They will make crystal violet staining strong. Okay, the next step is very important. We're using 95% alcohol to do decoloration. These 2 steps, basically we do 30 seconds. Decoloration. Because alcohol could be moving the dye or washed off the dye, so we do very short, only 10 seconds. How you count 10 seconds? Just count 10 Mississippi. 30 seconds, how you do it? Some people say sing a happy birthday song. but we usually don't do that. We just time in 30 seconds. Last one, we add saffron. This is also, we call it a counter stain. So this is very important, the procedures. And in the lab section, I always talk to them. The procedure is important. If you messed up the procedure, it will not show the ideal results. But the timing is not important. You can just staining 45 seconds, staining a minute, that's okay. The only critical thing is that 10 seconds decoloration process is critical. Okay, next question. Why gram-positive stains purple and gram-negative stains pink? So that's the principles. That's the principles behind that. First of all, you need to know gram-positive stains purple or blue color. It is actually crystal violet color. Gram-negative stains pink, staining pink. It is a saffron in color. This is the first thing you need to know then before we move on to the detail. So what's the principles? Basically, there are two theories about it. Number one, cell wall theory. This is a gram-positive cell wall, what it looks like. And I always draw my cell. This is needs a little bit of background of biology. What this one looks like, phospholipids interreacted with protein in a movable equilibrium balance status. This is called fluid mosaic model. And we know that every single phospholipids, it is hydrophilic head and hydrophobic tail. What means hydrophilic? Like water. What is a hydrophobic? Dislike water. We don't like water. So what's the example? Can we add oil into water? Are they going to be dissolved? No. The oil is going to be on the surface. The interface will not be directed. That's called hydrophobic. Can the sodium chloride dissolve in the water? Yes, that's hydrophilic. We want a hydrophilic because it's easy preparing. Okay. So gram-positive bacterial cell wall. Let's see what it looks like. There are very heavy area right. This is called peptidoglycan. Peptidoglycan, composed by modified sugar, NAG, NAM with amino acids. So in this class, we just briefly. In my other class, we talk about NAG, NAM structure, or lots of stuff. We just tell you, peptidoglycan, very heavy. Okay, this is we call it a periplasmic space. The outside there is some other things called teichoic acids. The function is unknown. Okay. Now, what's a gram-negative looks like? It's very interesting. You see the difference. Gram-negative bacteria, this area is the same. Fluid mosaic model is always the same, so you don't have to really worry too much. What's the difference? They have peptidoglycan. It is very thin. Thin peptidoglycan. Very, very thin, extremely thin. And above, what are you going to see? You have another layer. Yeah, you have another layer. This is Freud mosaic model again. Then what you have is LPS, lipopolysaccharides. Lipopolysaccharides. LPS. There comes out with cell wall theory. What does it mean? Gram-positive bacteria have heavy peptidoglycan. So, you add crystal violet, it's purple color. You add a green gram iodine, stays strong. Then because of the heavy peptidoglycan, you do 95% alcohol decoloration, it's not going to wash off. Prevent decoloration by alcohol. However, gram-negative bacteria, because of its very thin peptidoglycan, they cannot hold on the crystal violet during decoloration process. Okay? Gram-negative bacteria, very thin peptidoglycan, cannot hold on crystal violet. That's a reason. This is a reason. Lots of the textbook are written about that. That's called the cell wall theory. But the second theory I want to mention is lipid theory. What the lipid theory means? For gram-positive bacteria, only 5% lipids. For gram-negative bacteria, at least 10 to 15% lipids there. Because this guy on the top is lipids A. That's why we call it LPS, lipopolysaccharides. Lipopolysaccharides is a very magical stuff for gram-negative bacteria because they will generate endotoxin. That's the reason E. coli, Salmonella, all these gram-negative bacteria is a foodborne pathogen because of lipopolysaccharides. It will generate endotoxin. So what's the story here? I add iodine there. I add a crystal violet iodine. That's good. Stays purple. I have that high amount of the lipids. And decoloration, I use alcohol. Remember, lipids will not dissolve into water, but lipids is very good to dissolve in the alcohol. Is that right? So lipids dissolved well in alcohol. This is very easy knowledge. If you're preparing a meat, your hands are very sticky. What you do? Just dumping your hands with a little bit of wine. It's going to wash it off. Of course, obviously, the easiest one, you use soap and hot water. But if you don't have it, just dump a little bit of vodka or whiskey. Then it's going to go. The oil is going to go. Because it's dissolved well in the alcohol. So the lipids with these dye dissolve in the alcohol, which means the decoloration is well operated. So what happened? At that moment, it is blank. There is no color there, ideal. So we're gonna counter stain. Add the saffron name. It's gonna show pink color there of saffron. Okay, so that's the whole story. Okay, so that's what we have today for the gram stain. We will review this again at the beginning of next class. So we move to talk about the acid fast indole spot stain and all the others.